Hello, this is Dr. Morris. I'm starting a lecture series on epidemiology. This is chapter 14 in your textbook, and also chapter 14 epidemiology on course documents on Blackboard. The principles of disease and epidemiology. Basically, we're susceptible to a number of pathogens. Pathogens are referred to as disease-causing microorganisms. Pathology is the science that deals with the study of these diseases. This involves an etiology or a cause of disease. Now, in clinical settings, you'll hear the term etiology referred to quite a bit, so it's important that you understand the definition of etiology, which refers to the cause of certain diseases. Now, we're specifically talking about microorganisms as the etiology behind the diseases we'll be talking about in microbiology, but recognize not all diseases are caused by microorganisms. We'll also make reference to the Koch postulates, and if you recall from the Koch postulates, that refers to diseases that are caused by microorganisms. It does not relate to diseases caused from other environmental exposures such as toxins or any other diseases such as certain cancers, diseases such as kidney failure, heart disease. Heart disease, particularly coronary artery disease, blockages of the artery, is not related to a microorganism. So remember, when we refer to etiology as far as microbiology, we're referring to microorganisms being the cause of the disease. The manner in which diseases develop is referred to as pathogenesis, and then the structural and functional changes brought about by the disease and the final effect on the body. Infection means invasion or colonization of the body, the host in this case by potentially pathogenic organisms. Disease itself is any change from the state of health that is a normal or abnormal state in which the body is not properly adjusted or capable of performing its normal functions. So the terms in review: pathology, study of disease; etiology, study of the disease or the cause of the disease; pathogenesis, the development of the disease; infection, referring to colonization of the body by pathogens; and then disease, an abnormal state in which the body is not functionally normally. Microorganisms that establish permanent residence without producing disease are known as normal flora or normal microbiota. As you recall, we have discussed normal microbiota or normal flora previously in the chapters and in other discussions. Other microorganisms that may be present at the time and then disappear are referred to as transient microbiota or transient flora. Normal microbiota can benefit the host by preventing overgrowth of formal microorganisms, a process referred to as microbial antagonism. An example of production of bacteriocidins or bacteriocins by E. coli cells in the large intestines inhibits pathogens such as Salmonella and Shigella. So it's important to recognize that normal flora can also help prevent other diseases from occurring, even though normal flora or normal microbiota or microorganisms, these are typically organisms that don't necessarily cause disease and in fact will help prevent a number of diseases as well. The relationship between normal microbiota and healthy persons, one referred to as symbiosis. If in symbiosis one of the organisms is benefited and the other is unaffected, the relationship is known as commensalism, and you do need to know the definition of commensalism. If both organisms are benefited, it is called mutualism, and if one organism is benefited at the expense of others, it is referred to as parasitism. Okay, under certain conditions, the relationship can change. Members of normal microbiota can become opportunistic pathogens. In these particular cases, these organisms will lead to disease. In addition to competition among microbes, again, the symbiotic organisms or the normal flora compete with disease-causing organisms such as Salmonella, Shigella were the examples that were given. So when these organisms compete, when you see competition there, there's a situation of cooperation among microbes, and this becomes a factor. For example, independent organisms or molecules may be cofactors that synergistically enhance pathogenicity. Synergism means that two microbes or two microorganisms acting together have a greater effect than either acting alone. An example of this is a cultured cell infected with Mycoplasma bacteria. They are more susceptible to death from viral infections. Transient microbiota may be present for days, weeks, or months. Normal microbiota permanently colonize the host. Symbiosis is a relationship between normal microbiota and the host. So just as a review of the definitions that we've talked about. In commensalism, one organism is benefited and the other organism is unaffected. In mutualism, both organisms benefit. In parasitism, one organism is benefited at the expense of others, and some normal microbiota are opportunistic pathogens. Now, here are some of the locations that you would see normal microbiota in the host or in the human body. Locations include the upper respiratory system and the gastrointestinal system. The other area that we see a number of normal flora or normal microbiota would be the skin. So those are three primary organ systems: gastrointestinal tract, respiratory tract, and the skin, where you see the location of normal microbiota or normal flora. Normal microbiota in the relationship with the host, we see microbial antagonism, which is competition between the microbes. And again, normal flora or normal microbiota can protect the host from a number of diseases because it competes with the nutrient sources for the pathogenic organisms. So thus, by occupying the same niches that pathogens might occupy, we can see normal flora can prevent infection. Also, some of the normal microbiota produce acids. They produce bactericidins, which, if you remember from anatomy and physiology, and also earlier lectures in microbiology, you see bactericidins are produced by a number of cells and a number of body systems to help prevent infection. Most notably, in the male reproductive tract, there are glands that particularly produce bactericidins that help prevent urinary tract infections, particularly in the males. Probiotics are live microbes that apply to or ingested into the body, which are intended to exert a beneficial effect. There are a number of agents out now which will induce normal flora into the gastrointestinal tract. Individuals who may take antibiotics for a prolonged period of time and have unfortunately eradicated the normal flora that's in the gastrointestinal tract are sometimes given probiotics to actually induce live microbes into the gastrointestinal tract to reestablish the normal flora. Not all diseases are caused by microorganisms. We have inherited or genetic diseases like hemophilia or degenerative diseases such as osteoarthritis that are not related to infectious diseases or microorganisms. In these particular cases, this again refers back to the term etiology. The etiology here is not related to microorganisms. Here we'll discuss infectious diseases, however, and these are the diseases whose etiology truly is microorganisms as the cause. Referring back to Koch postulates again, as I alluded to earlier, it must be fulfilled in order to demonstrate that a specific microorganism is the cause of a specific disease. The rules are as follows, or the postulates are as follows. The same pathogen must be present in every case of the disease. The pathogen must be isolated from the disease of the host and grown in pure culture. The pathogen from the pure culture must cause the disease when inoculated into a healthy, susceptible laboratory animal, and the pathogen must again be isolated from the inoculated animal and must be shown to be the same pathogen as the original microorganism. Thus, you can see in Koch postulates that this is specifically related to microorganisms as the cause of disease. For example, a few diseases, such as syphilis, are caused by organisms that cannot be isolated in virulent strains grown in laboratory media. Now, this would be the exceptions to the Koch postulates, but this is still related to microorganisms, but this particular microorganism doesn't follow the postulates. However, evidence based on the first postulate over any year's experience has often been adequate to show that certain organisms is associated with the disease. Some diseases have such a clearly defined symptom, such as diphtheria or tetanus, that associations between specific microorganisms and the diseases are obvious. Other diseases, such as pneumonia, peritonitis, meningitis, may be caused by a variety of microorganisms, and specific etiology is not easy to determine from symptoms. Furthermore, some pathogens may infect a number of different organs or tissues and may cause very different diseases or disease symptoms. A good example is Streptococcus pyogenes, which can cause sore throat, scarlet fever, erysipelas, and puerperal fever. Cost postulates, again, just as a review, used to prove the cause of infectious disease. Again, infectious disease is caused by organisms. This illustration to your right is out of your textbook and shows that microorganisms are basically isolated from a dead animal. Colonies are formed on the culture plate. The microorganisms are then grown in a pure culture, and then after they've been identified in a laboratory setting, the microorganisms are then injected into the animal. The disease reproduced in the second animal. Microorganisms are again isolated. These are the pathogenic microorganisms, which are then grown in pure culture, and then the identical organism should be able to be identified in the lab under microscopy. So how do we classify these diseases? Well, there are some terms that you need to be familiar with, the first of which is symptoms, which are changes in the body's function felt by the patient, such as pain, malaise, a vague feeling of body discomfort that are subjective and not apparent to an observer. The patient also may exhibit signs. This is objective changes that the physician can observe and measure. And then a specific group of symptoms or signs accompanying a particular disease are called a syndrome. Should be familiar with those definitions. Also, the following definitions are important to know as well. Communicable diseases are the spread directly or indirectly of one host to another. Typhoid fever, tuberculosis are good examples of that. And non-communicable diseases are caused by microorganisms such as Clostridium tetani, which only produce tetanus when it's introduced into the body or by a contaminated wound. And then a contagious disease is easily spread from one person to another. Some other terms that are important, the term incidence and prevalence. The incidence of a disease is the fraction of a population that contracts it during a particular length of time. The prevalence of a disease is a fraction of a population that has the disease at a given time. And then the term sporadic. If a disease occurs only occasionally, it's referred to as sporadic. When it is constantly present, as is the common cold, then it's referred to as being endemic. If many people in a given area acquired certain diseases in a short period of time, this is referred to as an epidemic, and we see that a lot during the specific influenza season. A worldwide epidemic is referred to as a pandemic of disease. So again, terms in review: symptoms, a change in body function that is felt by a patient as a result of a disease; a sign, a change in the body that can be measured or observed as a result of a disease; and then a syndrome is a specific group of signs and symptoms that accompany a particular disease. Communicable diseases, a disease that is easily spread from one host to another; contagious disease, a disease that is easily spread from one host to another; and non-communicable disease, a disease that is not transmitted from one host to another. Now, other terms: acute versus chronic disease. Acute disease is one that develops rapidly but lasts only for a short time. Influenza is a good example of that. A chronic disease develops more slowly, and the body's reaction is often less severe, but is continuous or reoccurrent for long periods of time. Tuberculosis, syphilis, leprosy, hepatitis are examples of these types of diseases. Diseases intermediate between acute and chronic are referred to as subacute diseases. A latent disease, also another term that's important for you to recognize, is one in which the pathogens is inactive for a time, but then it becomes active to produce symptoms. Shingles is an excellent example of this. When many immune people are present in a community, and again, remember, these immune people don't necessarily present signs and symptoms of the disease. So when many immune people are present in a community, herd immunity exists. That is, susceptible persons are so few that communicable diseases do not cause an epidemic. These are the terms, again, just in review, that you'll need to appreciate: incidence, prevalence, sporadic disease, endemic disease, epidemic disease, pandemic, and herd immunity. You do need to be familiar with these terms and recognize their definitions and the importance. Again, other terms: acute, chronic, subacute, and latent diseases, all important, and you should recognize these definitions as well. Now, what extent can infection involve the host? Well, individuals can suffer from a local infection. This is one in which the invading microorganisms are limited to a relatively small area of the body, as for example boils or abscesses. In systemic or generalized infection, microorganisms or their products are spread throughout the body by the blood or lymphatic system. A focal infection is one in which a local infection, such as infected teeth, tonsils, sinuses, enter the blood or the lymph and spread to other parts of the body. The presence of bacteria in the blood is known as bacteremia. The rapid multiplication of bacteria in the blood is called septicemia or sepsis. Toxemia is the presence of toxins in the blood, and viremia is the presence of viruses in the blood. Again, terms that you should be familiar with: local infection, systemic infection, focal infection, bacteremia, and septicemia. Now, primary versus secondary infection, other terms that you need to be familiar with. A primary infection is an acute infection that causes an initial illness. A secondary infection is one caused by an opportunistic only after primary infection has weakened the body's defenses. A subclinical infection, or an inapparent infection, is one that does not cause any noticeable illness. Again, terms in review: toxemia, toxins in the blood; viremia, viruses in the blood; and then the terms primary, secondary, and subclinical disease you do need to be familiar with. Predisposing factors. Predisposing factors make the body more susceptible to disease. Short urethra in females, inherited traits such as sickle cell genes, climate, weather, fatigue, age, lifestyles, and/or chemotherapy all can affect the body's ability to fight off infection, and thus will make the body more susceptible to certain diseases. These are the stages of diseases. Should be somewhat familiar with this graph. Again, look at the X and the Y intersecting lines. The number of microorganisms, the time interval involved, helps to determine incubation period, prodromal period, periods of illness, the death if immune response and/or medical treatment fail, period of decline, and then period of convalescence. Notice at the peak is when death if the immune response and/or medical treatments fail. And again, the load of microorganisms typically at this point is at its highest. It's important to recognize these stages of disease and illness. Another term that's important to know is reservoirs. Reservoirs is a continual source of pathogens, such as an animal or an inanimate object, which is a reservoir of infection. Many people harbor pathogens and transmit them to others directly and/or indirectly. These people may be diseased and obvious transmitters, but others, called carriers, do not exhibit the symptoms. Diseases that occur primarily in wild and/or domestic animals, but they can be transmitted to humans, are called zoonosis. Also an important term to be able to recognize. Transmission may be direct contact with infected animal, contamination of food, water, insect vectors, or contact with contaminated hides, furs, or feathers, or consumption of infected animal products. Examples of non-living reservoirs for infectious disease are soil, which harbors agents of botulism, and water contaminated by human or animal feces that transmits gastrointestinal pathogens such as Shigella, and forms of dysentery. Okay, reservoirs. Reservoirs of infection are continual sources of infection. In humans, AIDS and gonorrhea. Carrier may have inapparent infections or may have latent diseases. Cases of HIV disease and AIDS, and cases of gonorrhea, once they pass that initial stage, you may not recognize any signs and symptoms of the disease in the individual that's infected, but he would still be a reservoir of the disease. Animals that house rabies, Lyme's disease, these are zoonoses that may be transmitted to humans. And then non-living sources or non-living reservoirs would include botulism, tetanus, which exist in the soil. Now, what is the modes of transmission that we can see? Contact transmissions. Infection can be spread more or less directly from one host to another by direct contact, such as kissing, handshaking, bites, or sexual intercourse. droplet infection in which agents of disease are spread very short distances, less than a meter, while contaminated in droplets of saliva or mucus from coughing or sneezing, also is considered to be a form of contact transmission. Indirect contact transmission involves a non-living object such as drinking cup or towel called a fomite. Vehicle transmission: inanimate reservoirs such as food, water, or blood can transmit disease. Vehicle transmission to large numbers of individuals. Disease is spread by agents of infection traveling on droplets or dust for a distance of more than a meter are considered to occur by airborne transmission. Spores produced by fungi can also be transmitted by an airborne route. Okay, I'm going to stop the lecture here now. This will end part 1 of the epidemiology lecture. The next part will be part 2, and we'll start with vectors at that point.